Hi everyone, today we're going to be talking about cellular communication and the different pathways that a cell uses. The first pathway we're going to talk about is the G protein pathway. Some important vocabulary to know for this pathway is the G protein linked receptor, which is the attachment point for the ligand. The ligand, which is a signal molecule that binds to the receptor protein. The G protein, which acts as a relay protein to carry the messages. Phosphorylation, which is a process that turns on the G protein, and the G protein pathway itself, which is the most common pathway used by cells. First, a ligand binds to the G protein linked receptor, which causes a shape change. This shape change causes phosphorylation, which turns on the G protein and causes GTP to switch out with GDP and bind to the G protein. When this occurs, the G protein splits into two subunits, the alpha and the beta subunit, so that they can go and activate other proteins. The G protein binds to the target protein and activates it. When this process is finished, the alpha subunit is hydrolyzed to GDP and the beta subunit is deactivated. These two subunits rejoin to form the inactive G protein, and that way the process can begin again. The next pathway we're going to talk about is the tyrosine kinase pathway. This pathway transfers phosphate groups from ATP to tyrosine molecules, which is another protein. The signaling molecules, or the ligands, bind to the extracellular domain of two nearby tyrosine kinase receptors. The two receptors bond together or dimerize. Phosphates are added to the tyrosine residues on the intracellular domain of the receptors, which is basically just another way to say phosphorylation. The phosphorylated residues can then transmit the signal to the next messenger within the cytoplasm. This pathway is then able to trigger a cellular response. The tyrosine kinase pathway is mostly involved with processes such as growth or emergency repair, and it also acts as a catalyst for rapidly activating up to 6 relay proteins at a time. Alright, the next pathway we're going to talk about is the ion channel receptors. Some vocabulary to know for this is the ligand-gated ion channels, which act as controls for particular signals. These ion channel receptors bind a ligand and open a channel through the membrane that allows specific ions to pass through. So when the ligand binds to the closed channel, it opens up, and specific ions are then able to pass through to the other side into the inside of a cell. Many of the amino acids in the membrane-spanning region are hydrophobic in order to interact with the phospholipid tails. However, the amino acids on the inside are hydrophilic in order to allow water and ions to pass through. When the ligand binds, there's a conformational change which allows the gate to open up and the ions to pass through. Ions like sodium, calcium, magnesium, and hydrogen are then able to come through and start depolarizing the inside of the cell in the cytosol. The next pathway that we're going to talk about is the intracellular receptors. Some important vocabulary to know for this pathway is the transcription factors, which start the making of mRNA within the nucleus. The intracellular receptors are mostly for receiving hormones and steroids. These molecules are lipids, so they don't need receptor proteins on the cell membrane. They're able to diffuse directly through the phospholipid bilayer. They are also hydrophobic molecules, which allows them to be able to do that. These molecules bind to proteins that act as regulators of mRNA synthesis, which is transcription, to indicate gene expression. Gene expression is the process of transforming the information in a cell's DNA into a sequence of amino acids forming a protein. When the ligand binds to the internal receptor, a conformational shape change is triggered that exposes a DNA binding site on the protein. The ligand receptor complex moves into the nucleus, then binds to a specific regulatory region of the chromosomal DNA and promotes the initiation of transcription. Intracellular receptors can directly influence gene expression without having to pass the signal on to other receptors or messengers. The next thing that we're going to talk about is the secondary messengers, which includes cyclic AMP. For this messenger, the ligand binds to the G protein receptor. When this happens, the G protein is able to bind to this, which also changes the shape of the receptor protein. The G protein goes to bind to this protein, which is able to make ATP. The ATP is able to change into CAMP, or cyclic AMP, and is able to produce all of this cyclic AMP. In turn, the cyclic AMP is able to bind to things like inactive kinases, which are then able to make active kinases, like that. Finally, we're going to talk about the secondary messengers, calmodulin and inositol triphosphate. The secondary messengers are important because they help to relay and amplify signals from the cell surface receptors to trigger a cellular response. The calmodulin and inositol triphosphate specifically are involved with initiating and creating muscle contractions. The ligand it comes and combines with the receptor protein. The shape change occurs in the receptor protein. And the shape change allows for the inactive G protein to attach to the receptor protein and become phosphorylated by ATP. This activated G protein will then travel to the inactive PIP2. The G protein will break a bond in the PIP2 to convert it to an active state molecule called IP3. The IP3, or inositol triphosphate, will then travel to the endoplasmic reticulum of a muscle cell. The endoplasmic reticulum acts as sort of a storage facility for the calcium ions, or the calmodulin, because calmodulin has a very low concentration in the cytosol, and the ion pumps in the plasma membrane work hard to remove it, which is why it's stored there. To open the endoplasmic reticulum and let the calcium ions into the cytosol, the IP3 binds to a receptor protein in the endoplasmic reticulum. This causes a shape change to occur in the receptor protein, and the shape change is what allows the calcium ions to flow out into the cytosol in the form of calmodulin. This calmodulin, which is also a secondary messenger, is able to then go and attach to the myosin microfilaments, which is what initiates muscle contractions.